almost good evening to you guys. And thank you very much, Anna, for this opportunity. My name is Jurandir Magalhães. You can call me Jura. And I'm a research geneticist at Embrapa Maize in Sorghum, working on sorghum for over 2 decades already. So it's really a great pleasure to be here to this seminar series, Crops to End Hunger, Case Studies in Africa and Beyond. So this is very timely. So we now see a lot of awareness of this problem in Brazil again. So this is very important. So our government is again concerned with this problem, which is quite real for sure. Today I have the pleasure to introduce to you Dr. Andekelili Mwamahonje from Tanzania Agricultural Research Institute. And I'm going to say a few words about Dr. Mwamahonje, and then I'm going to say just a little bit. Everything okay, Anna?
Yes, I have to mute some persons.
Okay,
Yes.
that's good. Let me start just by giving you a little bit of a background on Dr. Mwamahonje. So Dr. Mwamahonje has a Master of Science in Sustainable Agriculture at the Nelson Mandela African Institute of Science and Technology in Tanzania, and then a PhD in plant breeding at the University of Ghana. And the focus of his work is on molecular and conventional plant breeding of cereals and grapes as well, for tolerance to biotic, antibiotic stresses as well, but focusing on tropical regions, okay? Just a few words about that. I mean, when Anna invited me to make this presentation, I started to wonder, I mean, why does it feel special to me to introduce an African scientist? And I kind of started thinking about the reasons for that. And the reasons are actually very, very clear and logical. There are just very strong links between Brazil and Africa. So there's very strong cultural ties. We see these everywhere in Brazil. And it's because Africa is really the major founder of the Brazilian nation. Right. And we are increasingly recognizing these as we grow as a mature country, as a developed country. We're not completely there yet. Sometimes we see some setbacks, but the path has been laid, and I think we are just recognizing this foundation and being very proud of it. So I think that's the reason why it feels familiar and very pleasant to introduce to you an African scientist. So it's kind of like introducing a brother, if I can put it that way. Okay, so I talked about cultural ties between Brazil and Africa, which are obvious, but the ties are not only cultural. We also see very strong environmental ties between Brazil and Africa. So we both have widespread oxy soils, tropical soils in general with low pH, with aluminum and iron oxides in the clay fraction. So we have many abiotic problems for crop production on those areas. And then I'm going to talk a little bit about the work we do at Embrapa Maize and Sorghum. And we're really taking sorghum production on tropical regions as a multi-factorial problem, because we know that there's not a single problem. We have problems with aluminum toxicity, and aluminum is really a very elusive problem because we can find aluminum, ionic aluminum, very deep in the soil profiles, and our soils are very old. Aluminum toxicity is going to have a strong indirect effect in drought tolerance as well. Not only drought, but absorption of other nutrients, because it's going to damage the whole root system. So this is a problem that we have been working on the basic level and at the applied level for many years. And now we have resources to help breeders do marker-assisted selection, marker-assisted introgression, for example, for aluminum tolerance. And we do see a very strong effect of that. I mean, it could go to about 1 ton per hectare in the presence of drought. I mean, we always see aluminum tolerance in combination with drought. But we also have problems with phosphorus fixation into the soil clays. So phosphorus is highly unavailable on those soils. So it's really a very strong fight between the soil and the plant, and usually the soil wins this fight. So we are working on identifying genes that can modulate root system architecture and also morphology so that we can enhance phosphorus diffusion, the movement of phosphorus from the soil clays towards the root surface, so that it can be absorbed by the phosphorus transporters, high and low affinity transporters. And we have resources ready for that too. And lastly, we have also been working on drought tolerance. We're really looking at GBAE, the environmental causes in GBAE, and we are identifying genes related to drought tolerance, but not only drought tolerance. Now it's from our screens, it's more than clear that we have to consider heat stress in sorghum because it's going to reduce sorghum yields as well. So we see drought now in combination with heat, and we are identifying genes that can be used in sorghum by marker-assisted selection, or even in other crops by genetic engineering. So in terms of aluminum, we deployed the markers for a major aluminum tolerance gene into the EIB mid-density genotyping system so that people can use. And I'm going to be more than happy to give advice or help as much as I can. We have genes already validated for phosphorus acquisition efficiency, but we are working on many other genes as well, using DAR tag and things like that. So I expect for the next version of the mid-density genotyping system that we should have other markers to deploy into the system so that people can use it, but including drought. So this is all I had in terms of background for Embrapa, and now I'm going to take just a couple of minutes here to introduce you to the Global Sorghum Association. If you went to the last Global Sorghum Conference in Montpellier, you probably saw the official launch of the Global Sorghum Association, and I want to explain to you again here and just introduce for the new ones here what it is the association, what this is about. So we were launched in 2023 in Montpellier, and then our mission is very clear. We want to keep the Global Sorghum Conference happening at the highest level. So we had the first one in South Africa, and then the second one was last year in Montpellier, and then the third one is going to be in the US, organized by Texas Tech University. We are a formal entity incorporated in the US, and there is a reason for that. We want to be able to move funds from one host to another. We do not want this Congress to be just localized to a certain region. We really wanted to travel the globe. So I'm looking forward to see this meeting happening in Africa as well. So we're going to have a call for applications, and it's going to make me extremely happy to see this happening eventually in Africa as well. And there's no need to explain why. Our motivation, of course, is the Congress, but we want to try to use the Congress, which convenes the whole community, to build and strengthen the global sorghum community. And there is a reason for that. Our challenges are very complex, but sorghum is a key player, I mean, on agriculture in a climate change scenario because of its resilience to climate change, right? We all know about that. But we think that if we can explore, put the community working together, which is a big challenge, I think we can explore synergy and move faster into this new kind of scary scenario of global climate change. I'm the current chair of the initiating board of the GSA. We also have Dave Jordan from Queensland in Australia, Sarah Sexton-Bowser, she's from Kansas State. We also have Tim Lust from the US, the United Sorghum Checkoff Program, Tim Dalton from Kansas, and then we have Vincent Vades and Jean-François Remy from CGIAR, okay? But I want to tell you that this is just the initiating board, okay? Our idea is really to make the GSA a reflection of you. So we are going to have elections for the board, and this should happen this year. We're still setting up websites and just working on all this foundation of the GSA, but the elections are going to happen this year. You should be contacted. We would love to see you join the GSA and perhaps just putting forth your candidacy for the board as well. We want to make the Global Global Sorghum Association, a reflection of the whole community. I think this is the only way this is going to happen. Okay, so without further ado, I'm gonna go back here to my first slide. And the title, and I'm gonna again introduce Dr. Muhammad Honje, and the title of his talk is Introgression of QTLs for Drought Tolerance into Farmers' Preferred Sorghum Varieties. And I think he's gonna talk about stay green, which is really the most logical target if one is concerned with drought stress as well. I'm really looking forward to see Dr. Muhammad Honje's presentation. So with that, thank you very much, and we'll be in touch. Oh, and then I think I have to turn the word to you. Sorry about that, Nana. You have the word.
No problem. Thank you, Julandi. Thank you very much, and thank you for the announcement. So I think it's a very interesting announcement for all the ones that are here. So, Anda Khalil, can you share your screen, please?
Okay.
Not yet. Yes, thank you.
Okay. So, good morning, good afternoon, good evening, colleagues. My name is Andekele Saroli Mamahonje, a senior research scientist from TARI Mapora Center. I'll be presenting on the title Integration of QTL for Drought Tolerance into Farmers Prefer Sorghum Varieties using CASIP SNP markers. So my presentation will go through introduction, objective, material method, results, conclusion, then implementation. To start with, so globally the sorghum ranked as the fifth type of food crops after maize, rice, wheat, and barley. And then the global production is estimated to be 59.3 million tons. And the major producing countries globally is USA, next by Nigeria and India. And over 100 million people in East Africa depend on sorghum as a source of food. Tanzania produces an estimate of 800,000 metric tons annually in more than 10 regions. Most centered the center part of Tanzania, Lake Zone, and the southern part. So has different uses. For instance, it has potential to prevent diabetes, cancer. It also improve bone health, but also improve digestion and the iron, which variable in that crops reduces anemia. But the crops is also used as stock feeding, but it's also used for processing alcohol, among uses. So sorghum is a C4 crop which is characterized by drought tolerance, and drought is among the major constraints of sorghum production globally. It causes 50 to 60% yield loss that lead to food insecurity in Sub-Saharan Africa, in Asia, and in Tanzania. Drought is common in central zone where the productivity is about 1 ton per hectare. In Tanzania, breweries have increased at least the demand. However, the stability is not consistent. Improved sorghum varieties have been developed, but have since lost uniformity and often succumb to drought stress. So breeding for drought tolerance can achieve by different approaches like genome-wide association, conventional screening, marker-seed backcrossing. However, marker-seed backcrossing does efficient for integration rate of interest from the donor parent to the preferred recurrent parent. For instance, green 1, 2, and 4 have been mapped from the sorghum line B35 using a SSR marker, and recently stay green 3A and stay green 3B, which were mapped from the chromosome number 2 using a SNP markers. So these QTL have been integrated to non-stay green sorghum varieties for enhancing retention of stay green during post-flowering drought. Therefore, this QTL can also be integrated to the sorghum farmers preferred by Tanzania, just for post-flowering drought tolerant enhancement. So the general objective of this study was to develop drought high grain yield of sorghum genotype through marker seed-back crossing, and the specific objective were to introduce staging QTL from the donor parent B35 and A35 to the recurrent parents, and to identify the staging QTL to refer to the recurrent parent using CASIP SNP markers. So the study was conducted at field condition at Talim Center in Dodoma, Tanzania, but the genotyping was conducted in Sweden at the Intertech lab. So 2 donor parents and 9 recurrent parents this study were used, and the North Carolina 2 mating design was applied. In this case, where each of the donor parents were crossed with each of the recurrent parents to develop F1. This just to show the example, like the donor parent B35 crossed with the Marcia, the recurrent parent, develop F1, and this F1 were backcrossed with the recurrent parent Marcia to develop backcross 1 F1, and then the backcross 1 F1 were again backcrossed with the recurrent parent Marcia to develop backcross 2 F2. So this was done just for the whole recurrent parent. So the donor parent B35 was crossed with the recurrent parent Marcia to develop F1. So this F1 were backcrossed with the Marcia to develop backcross 1 F1, and then the backcross 1 F1 were again backcrossed with recurrent parent Marcia backcross 2 F1. So the backcross 2 F1 were planted for genotyping, where a total of 150 leaf samples by genotype were collected 47 days from planting for genotyping. So using theta SNP markers. So these are just a sorghum plants which are at 47 days from planting, where the sample was collected. And this is just a table which indicates the list of markers which were used for genotyping. And then the backcross 2 F1 were saved to backcross 2 F3, followed by genotyping with CASIP markers. Again, 150 leaf samples were collected per line. And then it was followed by DNA extraction, and then DNA purity. Again, amplification of DNA using SNP markers. So, and then this was followed by QTL identification for drought tolerance, and finally QTL selection. So backcross 2 F3 genotypes were genotyped using 10 CASIP markers. So why 10 markers? The backcross 2 F1 were genotyped using 30 SNP markers, but it was noted that out of 30 markers, 10 markers responded well. So these markers were just screened for genotyping backcross 2 F3. The marker data analysis was performed using SNP view from Biosearch technology, and subsequently were subject to flapjack. The phenotypic data of 2 F1 were based on the base 5 performing parameters, and in this case, the data were based on plant age, number of green leaves at maturity, panicle length, panicle width, and then influences exertion and grain weight per panicle, where for plant age, the highest was recorded from the parent F35, which were crossed with the patrol, and then next by donor parent B35 with NACOM TAMA1. And then for panicle length, the highest was recorded from the parent B35 crossed with the Y, and those with F35 crossed with the Hakika. And for panicle length, the highest was recorded from B35 from the parents B35 in NACO and then S35 with the pattern. And the highest gain weight per panicle was recorded from the process of the parent B35 NACO, and then the lowest was recorded from the donor parent B35. Based on genotyping result, 10% of the total sample collected for genotyping were heterozygous, and these heterozygous were identified by the SNP markers 49, 77, 102, and 103, which indicated heterozygous allele in about 37 samples of the donor parent S35 parent patro background. The rest 19 SNP markers showed homozygous allele for BACPOS 2F1 sample. So the selection of genotypes for advancing to BACPOS 2F3 population was based on the best 5 performing plants in response to genotypic data, where at least one samples were represented on the best performing samples. So table 3 indicates the favorable allele and staging identified after genotyping BACPOS 2F1 population, where the marker 39, 40, 41, 43, 44, 49, 53, 76, and 77 were identified staging 3. And the favorable allele were A, C, T, and G. And for SNP markers 89, 93, 94, 95, 101, 102, 103 were suitable for identifying staging 3B. And then for favorable allele was C, A, T, again, and D, G. But they are all contributing to drought tolerance. You can see, like they were able to retain the staging and trigger programmed cell death. Physical location, it was based from 56 to 72. So table 4 indicates the BACPOS 2F1 sorghum population score with the heterozygous. And in this case, the selection to advancing to BACPOS 2F2 and then BACPOS 2F3 was based on the samples which scored with the heterozygous allele. Like I have said, for the donor parent F35 and patro as the recurrent parent indicated the highest number of heterozygous allele. But again, this at least one sample were selected from each of the crosses, or from each of the donor parent and recurrent parents. So figure 2.1 and 2.2 indicate the phenotypic results based on the field evaluation of BACPOS 2F3, where the mean gain weight per panicle across the environment was highest in the genotype NA316. Just to say a little bit that after genotyping, so the sample were planted under field condition, under two environment, irrigation and the stress condition. So at least to see the interaction among the two environments, so the mean grain weight per panicle across the environment was highest for the line NA316. And then for the staging was highest for the donor parent, B35 with close to 4.5. And then the lowest was recorded from NA307, but almost there was some improvement compared to the recurrent parent. Again, out of the SNP markers used for genotyping backcross to F3 population, identified favorable homozygous allele for stay in sorghum. As I have said that 10 markers were screened from 30 SNP markers. Again, that 10 markers which were used for genotyping the backcross to F3, 2 did not express 8 markers. It indicated were able to identify some homozygous allele from the samples. As for instance, for the genotype now, NA241 were able to be identified with SNP marker number 72, 89, 102, but the highest was the SNP marker 89, and then followed by 102, and the lowest for this genotype was 72. So the rest were in between, except for the recurrent parents indicated the lowest number of favorable allele compared to the crosses. So in this case, that it shows that the process and the back process were successful, but the efficient differed among the parents which were used. Some of the parents, current parents, the expression was high, but some actually the situation was not as expected. So the further screening, so that at the end we come up with a new genotype which we can develop into line and then advance, at least propose for release as a new variety. And then table 6 indicates the favorable allele in the stage green identified in backcross to F population of sorghum. For this case, we were considering the stage green 3A and stage green 3B. So the marker 42, 49, 53, 54, and 72 identified stage green 3A, and the marker 89, 101, 102 identified stage green 3B. But if we compare between the two, those which identify 3A and 3B, there were higher number for those which identify 3A compared to those which identify stage 3B. Although those which identify stage 3B was high in terms of number of favorable allele, like you can see this 89 indicated 700 and 700 and then 728, and then the lowest was SNP 102, which indicated 727. But for those which indicated higher number of markers which identify stage in 3A, Kasip SNP number 42 only indicated 2 favorable allele compared to the rest 4. And in most cases, the physical location was based from 50 on the physical location was based on 56 to 72. And in the function of all these markers, we are expressing the trait of state delineation and nitrogen mobilization, and then programmed cell death, which together have contribution to stage green stabilization in the sorghum crop. So by concluding, so marker-assisted selection was performed in backcross to F1 and backcross to F3 population, and the QTL stage green 3A and 3B associated with stage green characterized from the donor parent B35 were successfully introduced into the two farmers' preferred recurrent variety, namely Nakomtaman and Seguifa. As you may remember that the donor parent F35, these were not progressive because we are not able they are not able to identify some of the targeted stay. Why? So we decided to stop it and then to progress with promising those which were promising results. So 3 genotype NA307, NA316, and SE438 outperform the recurrent parents Nakomtama1 and Seguifa. And the SNP markers 42, 49, 53, 54, 72, and 89 were associated with Seglin 3A. And then the SNP markers 101 and 102 are linked with Seglin 3B in the Bicross 2 F3 population. So the SNP marker which were linked with stage including the Bicross, in this study can be used as a reference in East Africa and abroad in the marker set back crossing for enhancing drought tolerance in sorghum. So recommendation. So further mapping of SNP markers which are tightly linked with stage QT should be done for enhancing drought tolerance in sorghum. So based on our research, so we recommend that a further mapping should be done because out of 30 we tested, only 10 works. So it seems there's something to be done more so that we are able to find those markers which can really point out or detect the stake in QTL. Again, the sufficient back crossing is again necessary to widen the chance of finding the targeted trait. So if you conduct a minimal number of or insufficient number of back crosses, it is possible that you may not be able to integrate short plants was a donor parent, the donor parents B35, and the tall one was a Nakomtama1. So these are the ones used to develop the NA307, which at least we are still continuing for multiplication evaluation, and it's performing well. We are evaluating now at the PVS, participatory variety selection. So if we go to farmer and if farmer will accept, then we can go further for registration as a new variety. So thank you.
Thank you, Andakelik, and sorry for the connection. Andakelik will share all the presentations, so all of you can after have access to the presentation. But now let's go for the questions. Bodo, do you want to ask your question, please? Unmute yourself.
Yes, hello. Hello, Anna. Hello, Dr. Andakelik. Thank you for this explanation on the, I think it was more on the genetics, molecular biology work to map these or to identify these SNP markers that can be used for marker-assisted introgression. I was interested in the phenotype of this stay green. Is it one trait or is it a composite trait, or what is it, and how it how is it detected in the field, or how is it stored? This kind of things. Maybe you can allude a little bit on that, or maybe the people from the Sorghum community may also add something for the rest of the audience to understand. Thank you.
Thank you for your question. And yes, it is true that it's very important to explain about this, that the phenotyping, we used a scale. There's a scale which was developed by EC Research. There was a scale which was developed by EC Research, and the scale is just 1 to 9. So we use the scale from 1 to 9, and then we usually evaluated after stressing condition, because this trial was based on the irrigation. So irrigation and stress environment. So the data were collected from this when the trial was stressed, and then we started just trying to check the greenness of the plants, and the data were taken during noon, where there is sun, so that we were able to differentiate between one line and another. So, for instance, one, it indicated that when the when the leaf is fully green, and when there is not any deviating color from a greenness. And then 3 indicated that the leaves is green, but with somehow with some other color or deviation of things from the greenness. But for 5, it means that the plant leaves is green, but it half 50 of the plant leaves in the plant is just green, but the rest are deviating from the green color. Maybe you find there is a sign of dryness, or maybe some kind of blight, or some kind of maybe yellowish. So that one indicates maybe those leaves could have been affected with a stressed environment. So we also compared with the donor parent, and the donor parent actually were almost green most of the time. But as the time goes, and when the stress continued, the situation went down. The greenish tend to reduce. But the other way we use to identify the green, we use the chlorophyll meter to measure the chlorophyll content from the leaves. So we measured at the top, at the middle, and at the bottom. We also tried to quantify the chlorophyll content. But for us, it also helped to know that which has more chlorophyll content compared to another. So it was another indicator that also were indicating that there is a relation with the staging on that plant. So among other factors we used, but this were the major 2 phenotyping tool we used. Thank you.
Thank you, Andrekeli. Bodo, do you want to ask the other question that you share in the chat?
Yes, yeah. Thank you, Dr. Andrekeli. So I understand from your description and from the slides you showed that this is a multigenic trait, and it seems that for the sorghum community, or at least for this program you have been working with, this group of genes is treated as just one lump sum reaction, which is called stay green according to a score that is applied by a scale. So for the future, if I were a breeder, I would be interested in seeing individual effects of individual SNPs, or let's say SNP-related unknown genes or whatever, what is there, to see whether some have a bigger effect than others and what nature these effects have. But this is only the other question I got from your last slide was, because you showed these two sorghum parents, you called them parents. So I was curious about the breeding system of sorghum. I understand this is a diploid crop. Is it diploid? And then is it self-compatible? Can it easily be selved? Is there inbreeding depression? Do you have homozygous lines in the end? And do you do hybrid development by crossing these lines? Or how are these parents described from the breeding system, from their genetics point of view? Maybe you can add something to that. Thank you.
Okay, so it is true. So first of all, so this is not a hybrid. We just develop a open pollinated varieties, not OPV. And it is true that for hybrid, and then you have to be very smart to avoid outcrossing. But yes, we use recurrent parent, which were again OPV, not a hybrid, because in Tanzania we have a number of varieties, which most of them are OPV. Of course, recently hybrid have started emerging, but the majority of varieties which are cultivated by farmers are OPV. So the recurrent, the donor parent which we requested from AgriSat also believe that it's a stable and well sustained line. But for the recurrent parents, which are varieties which are cultivated by farmers, are OPV. So for OPV, yes, there is some sort of maybe finding a mixed but they are not stable. So, but we try to at least to screen to select at least the stable variety. We try to minimize the off type, like those which were deviating from the normal height, for instance, and then maybe especially for plant height, at least plant height is one of the indicators which at least trying to show the stability of the variety. So we try to at least to remain with those plants which are same height, so that they were true to type. But I agree with you. Genetically, there may be some of the mixed up, but we try to escape that. And then for homozygous level, yes, to develop a line to homozygous, you have to control the environment really. For instance, I said we do saphing, so we make sure that at the booting stage, we try to use a saphing bait and try to cover the panicle, so that during flowering, so anthers from the pollen from the anthers does not get to drop, so that maybe it may outcross with others. We try to minimize that so that to avoid outcrossing. But still, there may be some of the outcross genetically, which for sure it may be difficult to control, but we try to control. But again, I can't confirm that maybe we did maybe 100%. No. But at least we try to maintain. And then if you see even in the field, you still able to see the variation of the recurrent parents. That means the farmers cultivated variety with those genotypes or line which we generated. So all those differences, and we have been testing in several times since 2019, 2020. So we see the difference. So that difference, and the stability of the difference between the parent and the genotypes, it at least give us assuring that we develop something which we can disseminate to our farmers. I'm not sure whether I have answered sufficiently, but that's what I can respond from your question. Thank you.
And Kelile, can I jump in? Anna, is it okay if I respond?
Yeah, it's okay.
To just add on to what Ande Kelile said. So your first question, Bodo, on the contributions of different QTLs to the phenotype, this has already been done for all the stay green QTLs. And specifically the one that Ande Kelile was talking about, stay green 3B, I believe the highest contribution that has been reported is 40%. I did a presentation here about maybe 2-3 months ago, where we were actually trying to look at the various SNPs from the same QTL and its contribution to drought, especially looking at it in drought tolerance. And we found 4 that were consistently, 100% favorable allele present in the different phenotypes. When it comes to specific candidate genes, the PIN genes have already been characterized by Borrell from Australia. This was found, the PIN genes were found to be consistent across, I think, at least 3 different stay green QTLs. And yeah, so these have already been characterized, but the contribution is of specific genes is quite low, usually like less than 10%. Obviously, there's need to understand the stay green QTLs a little bit more, because as I also mentioned in the previous presentation, we've only always looked at the above ground phenotypic data sets and not the below ground. So there are quite a number of efforts now looking at both above ground and below ground, just to be able to unpack the trait a little bit more, to be able to explain all the different morphological traits contributing to the phenotype. Secondly, yes, sorghum is a diploid. Sorghum is self-pollinating, and we do have hybrid sorghum being developed utilizing CMS. So I don't know if you were asking specifically in Andelile's project, which I don't know if that's what he did, but I don't believe that's what he did. But we do have hybrid sorghum production system that is well established using CMS. Yeah, if we're doing just, even when we're doing the regular hybridization, not particularly looking at CMS, we still bag our plants, otherwise you'll get contamination. So I don't know if that really answers your question. So the broader breeding program, we have sorghum hybrid production scheme, but even when we do OPVs, we still bag them because there's a significant amount of outcrossing that would affect the purity of your lines if you do not bag your selected lines.
Yeah, excellent, Damaris, excellent. I'm sorry I missed your talk you were referring to. I'm curious, maybe I can get the
It's available online, I think. Anna has always maintained them, so you can get it.
So Damaris' presentation is in 2023, is in the 3rd week. So it's available. So all the presentations are available, and this presentation will be included.
Very well. Thank you so much. Thank you, Damaris. Thank you,
Damaris. So there is any other question? If there is no other question, I think we can finish the seminar. Thank you very much to all. Thank you very much, Ande Kelile. I know the difficulties with the internet bandwidth. Thank you for being encouraged and come here and present your work. Thank you, Dr. Jurandir. Thank you very much. It was an honor. thank you to all. So we came to the end of one of the seminars. And we hope to see you in the next seminar focused on wheat, and will be presented by Dr. Susan Drezenkar from the CNIT. So thank you to all and stay well.
Thank you so much. See you guys. Bye bye.
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 Thank you.
Thank you.